Hey everyone, welcome to this explainer. Okay, today we are embarking on a pretty amazing journey. We're literally mapping out the actual blueprint of life itself. We're going to start way back with the core 19th century philosophy of what makes a living thing, and then we're zooming all the way in to look at the highly advanced, completely dynamic security systems of modern cells. Just to put this into perspective, an average adult human body, your body, is made up of around 30 to 40 trillion individual cells, trillions, and every single one of those cells functions like its own autonomous microscopic city, just humming along, keeping you alive. So if you've ever wondered about the invisible architecture that builds you, me, and literally every living organism on this planet, well, you're in exactly the right place. So our foundational blueprint for the entire explainer today, it actually comes from a single brilliant page of handwritten notes. It's this dense, totally information-packed cheat sheet that miraculously distills decades of really complex biological discovery into one clear framework. And honestly, rather than getting bogged down in heavy textbooks, we're just going to unpack these specific notes step by step. By the time we're done here, we are going to completely understand the fundamental unit of life. All right, here's our game plan for today. 1. The foundation of cells. 2. Prokaryotes and the cell envelope. 3. Internal and external modifications. 4. Eukaryotes and the cell membrane. And 5. We'll bring it home with the fluid mosaic model. Let's dive in.
Starting with Section 1, the foundation of cells, building the core philosophy.
You know, every single living thing on Earth actually relies on a set of historical rules. To wrap our heads around modern biology, we really need to understand the core philosophy that built it. So let's rewind to 1838. The scientific world back then was kind of like a wild frontier. Microscopes were getting better, but biology totally lacked a unifying theory. Then, a scientist named Schleiden figured out that plants are entirely made up of cells, which piece together to form tissues. In that exact same year, another scientist, Schwann, was looking at animals. He figured out they have a distinct outer layer, what we now call the plasma membrane. Schwann also noticed that the rigid cell wall Schleiden was seeing, those were completely unique to plants. So together, they combined their observations into one massive unifying theory. Everything, plant or animal, is made of cells and cell products. I mean, it was basically finding the biological unified theory of everything. But there was still a huge piece of the puzzle missing. Where were these cells actually coming from? And that brings us to 1855, when R. Virchow drops the modern addition to cell theory. Before this, you gotta understand, a lot of people genuinely believed in spontaneous generation, like the idea that life could just magically spring forth from dead matter. Maggots appearing on decaying meat, people thought the meat just created them. Well, Virchow completely shattered that paradigm. He delivered what is basically biology's ultimate mic drop moment, omnis cellula e cellula. Translated, this profound realization means that all modern cells come from pre-existing cells. Life doesn't just magically appear. It's a continuous, unbroken chain stretching all the way back to the very dawn of life on Earth. So every single cell in your body, it's the direct descendant of another cell.
Pretty wild, right?
Moving right along to Section 2, prokaryotes and the cell envelope. So moving past the historical stuff, how do the absolute simplest, oldest forms of life actually build their armor? Let's take a look at prokaryotes. These are the microscopic pioneers of our planet, and their ultimate defense mechanism is the cell envelope. You can think of this basically like incredibly sophisticated medieval armor made up of 3 distinct layers. On the absolute outside, we've got the glycocalyx. This layer is highly adaptable. It can either be a loose, sticky sheath, called a slime layer, which actually helps bacteria slip right past immune cells or stick to surfaces, or it can be a really thick, tough capsule designed to help the organism survive in extreme environments. Beneath that outer shield is the middle layer, the cell wall. This gives crucial structural support. Honestly, without it, the internal pressure of the cell would cause it to just pop and burst like an overfilled water balloon. Finally, the inner layer is the plasma membrane. This is the semi-permeable gatekeeper that actively interacts with the outside world. So that's 3 distinct layers working in perfect harmony to protect the delicate life inside. Next up, Section 3, internal and external modifications. Okay, so bacteria don't just stop at basic armor. They completely modify this simple envelope for their survival, adapting internally and externally to thrive basically everywhere. Let's start with the inside. Mesosomes are extensions of the plasma membrane into the cell. Now, that might sound super simple, like, oh, it's just a wrinkle or a fold in the inner lining, but it's actually a brilliant piece of engineering. By folding inward, the bacteria dramatically increases its internal surface area. And as we're about to see, these extensions basically act as highly complex internal machinery for a prokaryote that otherwise completely lacks complex organs. These simple membrane folds, the mesosomes, handle the absolutely vital operations that keep the cell alive. Take respiration, for example.
Bacteria obviously don't have lungs, and the simple ones don't even have mitochondria. Instead, they use the surface area of these mesosome folds to perform cellular respiration and generate energy. It's genius! These folds also help form the cell wall. They manage DNA replication and distribution when it's time to divide, and they facilitate complex secretion processes and enzymatic functions. It's literally a multi-purpose tool built right into the membrane. And it gets even more specialized. In blue-green algae, we see different plasma membrane extensions called chromatophores. These act like microscopic solar panels, packed with pigments that allow these simple organisms to actually photosynthesize sunlight into energy.
Now, externally, bacteria build completely different structures depending on whether they need to move around or stay firmly put. It's a perfect example of motility versus non-motility. On the motility side, we have flagella. These function basically like a microscopic outboard motor, playing a direct role in bacterial movement and propelling the cell through fluids. On the non-motility side, we've got pili, which are elongated tubular structures, and fimbriae, which are bristle-like. Now, these do not play any role in movement. Instead, things like fimbriae act more like grappling hooks or rock climbing gear. They're used specifically to anchor the bacteria onto rocks in a rushing stream, or, quite often, to latch tightly onto host tissues, which, fun fact, is exactly how certain bacteria cling to the back of your throat when you have an infection. Structure perfectly dictates function. Before we leave our prokaryote friends, we definitely have to talk about what's floating around inside their cytoplasm. Meet the inclusion bodies. You can think of these as the cell's pantry. Because a bacteria might suddenly find itself in an environment totally devoid of nutrients, it stores reserve materials right here. They keep stockpiles of phosphate granules for energy transfer. They store cyanophycine granules, which certain photosynthetic bacteria use to stash away nitrogen. They also keep glycogen granules as a backup carbohydrate energy source. But honestly, the most fascinating of all might be the gas vacuoles. These act like tiny buoyancy compensators, kind of like submarine ballast tanks. Found in blue, green, purple, and green photobacteria, these gas vacuoles inflate or deflate to help the organism float at the exact perfect depth in a body of water to capture the maximum amount of sunlight. How cool is that? All right, let's shift gears. Section 4, eukaryotes and the cell membrane. We are transitioning away from bacteria now to explore the advanced security system of complex eukaryotic cells. This is the exact blueprint of the cells making up your own body right now. Unlike the rigid armor-like bacterial walls we just looked at, the eukaryotic cell membrane is a living, highly dynamic barrier. It's a semi-permeable boundary made primarily of a lipid bilayer, specifically phosphoglycerides, studded with proteins and carbohydrates. And by semi-permeable, we mean it acts like an incredibly strict bouncer at an exclusive club. It doesn't just block everything, it actively decides exactly what molecules are allowed to enter the cell and exactly what waste products get kicked out. It's a highly regulated, bustling border crossing. The structural balance of this membrane is grounded right here in hard human data. If you actually analyze the cell membrane of a human red blood cell, you'll find this exact ratio: 52% protein and 40% lipids. This isn't just a random mixture. This highly specific balance is exactly what allows our red blood cells to be so incredibly flexible. I mean, they have to deform and squeeze through the absolute tiniest microscopic capillaries in our eyes and fingertips and then bounce right back to their normal shape. If this ratio of proteins to lipids were off by even just a little bit, the cell membrane would be too rigid, causing blockages, or too weak, causing the cell to literally tear apart under pressure. And finally, Section 5, the fluid mosaic model. So for decades, scientists knew roughly what the membrane was made of, but the actual mechanics of how it all fit together, that remained a total mystery. Earlier models treated the cell membrane like a static, rigid sandwich. But then, in 1972, scientists Singer and Nicholson proposed the fluid mosaic model, and it absolutely revolutionized cell biology. It states that the membrane isn't solid at all. Instead, because of the quasi-fluid nature of the lipids, proteins can actually move around within it. Imagine a vast sea of liquid lipids with massive protein icebergs freely floating, drifting, and bobbing right through it. It's literally a mosaic of constantly shifting pieces. This ability for lateral movement, meaning the proteins can seamlessly slide side to side across the surface of the cell, is the direct measure of the plasma membrane's fluidity. And it isn't just a quirky physical trait, it's a vital, measurable characteristic. The ability of these embedded proteins to shift, move, cluster together to form signaling complexes, and then drift apart again, that's exactly what defines how a eukaryotic cell can respond to a constantly changing external environment. To truly grasp the importance of this, we just have to look at what this fluidity actually enables. Basically, every major dynamic event in a cell's life relies completely on it. Without a fluid, flexible membrane, a cell physically couldn't expand and grow. It couldn't form intracellular junctions, the vital connections that hold neighboring cells together so our tissues don't literally fall apart. It couldn't secrete vital substances, like hormones or enzymes. It couldn't perform endocytosis, which is that Pac-Man-like process where the membrane physically folds around and swallows large nutrients from outside the cell. And perhaps most importantly, without this fluidity, cell division would be totally impossible. The very act of life continuing requires the membrane to gracefully pinch inwards and separate to create two entirely new cells. You are a living, breathing ecosystem made of trillions of these cells, forming a continuous, unbroken chain stretching back millions of years to that fundamental rule of omnia cellula e cellula. Take a second to just think about that. How does this incredibly delicate, microscopic, quasi-fluid barrier, constantly shifting and drifting in a perfectly balanced sea of lipids and proteins, act as the absolute fundamental boundary keeping you alive at this very second? It is a spectacular, breathtaking piece of natural engineering. Thank you so much for joining me on this deep dive into the blueprint of life. I really hope you leave this explainer viewing the world and your own body with a profound new sense of wonder. Keep exploring.